Bismillahirrahmanirrahim. We discuss biological functions of starch and biological functions of glycogen. First of all, key concepts. Key concepts include starch, structure of amylose, structure of amylopectin, properties of starch, and function of starch. After this, we will discuss glycogen, structure of glycogen, breakdown of glycogen, and last one we discuss functions of glycogen. So what is starch? Starch is a type of homopolymer. It is a carbohydrate. Starch is made up of only one type of sugar. The type of sugar is alpha D glucose. It is the main source of food for human being. It provides glucose molecules for the energy purpose. Starch is also known as amylin. It is an important food product and biomaterial used worldwide in many industrial sectors including foods, health, textile, chemicals, and engineering sectors, etc. Starch is homopolysaccharides made up of glucogenous. Starch enters the system of an animal or human by means of food. It is found in the cells of green plants, seeds of corn, wheat, beans, in the stems and roots of potatoes, and many other places. Starch is easily digestible when cooked due to its property of gelatinization. The chemical behavior of starch is dependent on the nature of its constituent compounds. Starch chain generally consists of 15 to 20% amylose and up to 80 to 85% amylopectin by mass. Some classification categorize starch containing less than 15% amylose as waxy, 20 to 30% as normal, and greater than 40% as high amylose starches. Amylose and amylopectin have different physiochemical properties which impact on the overall properties of the starch. It is often important to determine the concentration of each individual component of the starch as well as the overall starch concentration. The physiochemical properties of starch, including gelatinization and retrogradation, and functional properties of starch, for example, solubility, swelling, water absorption, syneresis, and rheological behavior of gels, determine the potential use of starch in the food industry. These properties depend on the molecular and structural composition of amylose and amylopectin, percent composition and arrangement of these two homopolysaccharides in starch granules. In this slide, we shown some starchy foods which we use in our daily life, such as bread, cereals, pasta, rice, potatoes, beans, and chestnuts. Amylose. Starch is made up of amylose and amylopectin. Now we discuss what is amylose. Amylose is unbranched, linear in nature. Amylose is made up of alpha-1,4 bound glucose molecules. The carbon atoms on glucose are numbered starting at the aldehyde carbon. So in amylose, the one carbon on one glucose molecule is linked to the 4 carbon on the next glucose molecule to produce alpha-1,4 glycosidic bonds. The number of repeated glucose subunits N is usually in the range of 300 to 3000, but these can be many thousands. The structure of amylose as shown below in the slide. Amylose is C1 and C4 linkage as shown in the figure below. First carbon is anomeric. Anomeric carbon is that carbon which is derived from the carbonyl carbon compound, and in the structure which is C1 is free and is called as reducing end. The opposite terminal is C4, which is not free and is known as non-reducing end. So it shows polarity in the form of reducing and non-reducing ends. The structural formula of amylose is pictured at below, in which one carbon of one glucose unit combined with the 4 carbon of other units to produce alpha link, and the bond name is alpha 1,4 linkage or glycosidic bonds. When we discuss or see the structure of amylose, it shows that glucose chain is straight line or straight chain, and it is fully extended structure. But this fact is not true. It is like a helical structure as shown in figure below. When our body metabolizes starch, it metabolizes the non-reducing end, and amylase enzyme acts on the non-reducing end. In contrast, amylose contains very few alpha 1,6 bonds or almost none at all. This causes amylose to be hydrolyzed more slowly but have higher density and be insoluble. Here we show the structure of amylose, in which alpha 1,4 glycosidic bond is shown in the structural formula of amylose. Now we discuss breakdown of amylose. Amylase is an enzyme acts on the substrates amylose which possess 3 or more than 3 glucose units. It means amylase acts on the starch, and as you know, amylase is an enzyme. Amylase cannot act on maltose because maltose is the 2 glucose molecular dimer. This is not the substrate of amylase. When this amylase is given enough time with the starch chain, it gives you maltose and dextrin. Dextrin is somewhat smaller polymer of alpha D glucose. Now the types of amylase. As you know, amylase is an enzyme, and there are 2 types of amylase. Number 1 is alpha amylase, and number 2 is the beta amylase. Alpha amylase is an important for humans. It is found in saliva and pancreatic juice. It is randomly cleavage and product is maltose and dextrin. Whereas beta amylase, it is found in the sprouted grains. It is cleaved in the non-reducing end, and product is maltose and limited of dextrin. Limited dextrin means only 4 glucose molecules are there. Amylopectin is also a part of starch. Amylopectin is a water-insoluble polysaccharide and highly branched polymer of alpha glucose units found in plants. It is one of the components of starch. Amylopectin is a branched polymer of D-glucose units linked by 1,4 and 1,6 glycosidic bonds as shown in the next slide. Glucose units are linked in a linear way with alpha 1,4 glycosidic bonds, whereas branching takes place with alpha 1,6 bonds occurring every 24 to 30 glucose units. Here we see the structure of amylopectin. The linear bond is alpha 1,4 glycosidic bond is present between one glucose unit of C1 and other glucose unit of with C4 carbon atoms. The branched bond is also available, which is known as alpha 1,6 glycosidic bond. Now, amylopectin is a much larger molecule with a higher molecular weight and heavily branched structure built from about 95% with 1,4 glycosidic bonds and 5% of 1,6 linkages of glucose units. Glucose units are linked in a linear way with alpha 1,4 glycosidic bonds, with the branching takes place with alpha 1,6 bonds occurring every 24-30 glucose units, resulting in a soluble molecule that can be quickly degraded as it has many endpoints onto which enzymes can attach. Its counterpart in animals is glycogen, which has the same composition and structure but with more extensive branching that occurs every 8 to 12 glucose units. Plants store starch within specialized organelles called amyloplasts. When energy is needed for cell work, the plant hydrolyzes the starch, releasing the glucose subunits. Humans and other animals that eat plant foods also use amylase, an enzyme that assists in breakdown amylopectin. Starch is made of about 80 to 85% amylopectin by weight, though it varies depending on the source, higher in medium grain rice to 100% in glutinous rice, waxy potato starch and waxy corn, and lower in long chain rice, amylo maize and russet potatoes. Amylopectin is highly branched, being formed of 2,000 to 200,000 glucose units. Its inner chain are formed of 20 to 24 glucose subunits. We discuss properties of starch. Critical properties are those properties exhibited without any change in chemical characteristics of starch and do not involve the breaking and creation of chemical bonds such as solubility, gelatinization, retrogradation and glass transition, etc. Chemical properties changes due to chemical reactions and usually involve the breakage and creation of new bonds. Examples of such chemical processes in starch include hydrolysis, oxidation, esterification and etherification. Research strongly indicates that molecular weight and branching attributes of starch, which play important role in the shape and size of granules, can potentially be used for predicting some of its functionality such as texture, fasting and retrogradation, etc. Amylose has more proportional relationships with fasting and gel texture properties, while amylopectin, which are predominant in regular and waxy corn starches, has higher proportional relationship with firmness. Now we continue properties of starch. Starch has some special properties which make it a unique natural polymer, such as thickener, rheology enhancer, film former, sticking adhesive, cohesive, water binding capacity, water retention, flocculent and shear stability. Solubility and gelatinization of starch. When unprocessed or native starch granules, which are relatively inert, are heated in the presence of adequate water, usually during industrial process, swelling of the granules occur and the amylose dissolves and diffuses out of the swollen granules, which upon cooling forms a homogeneous gel phase of amylose and amylopectin. The swollen amylopectin enriched granules aggregate into gel particles, generating a viscous solution. This two-phase structure called starch paste is desirable for many food applications where processed starches are used as thickeners or binders. Now, function of starch. Starch provides energy exactly like sugar. Nutrient defense unprocessed sources of starch containing more fiber and protein, which release energy at a more constant rate. Baked products, bread, pies, samosas, wafers, biscuits and sausages. Baked products like biscuits, pies, bread, cakes, wafers and sausages are high-density products requiring heat-resistant starches. Hence, cross-linked starches are used since they are more resistant to oven baking temperature of 120 to 230 centigrade. Gelatinized starches are also used in ready-to-eat cereal meals such as corn flakes, etc. The temperature, humidity and degree of stirring determine the texture and quality of the product. Confectionery, for example candy, sweets and sweetmeat. Oxidized starches have high clarity of transmittance, low viscosity and low temperature stability. It is frequently used in confectioneries for coating candies and sweets since they easily melt. Gravies, soaps and sauces, tomato paste or ketchup. Esterified and cross-linked starches are mostly used. Cross-linked starches have high stability for granules, swelling, high temperature resistant, high shear stability and acidic condition stability. They are used as viscosifiers and texturizers in soaps, sauces, gravies, bakery and dairy products. Esterified starches have improved clarity of starch paste, greater viscosity, reduced syneresis and freeze-thaw stability. Cross-linked starches are used in wide range of food applications such as gravies, dips, sauces, fruit pie, fillings and puddings. Mayonnaises, salad dressing, ice cream, spreads and beverages. Hydrolyzed and esterified starches are mostly used in salad dressings and beverages. Hydrolyzed starches, acid-modified starches has lower paste viscosity under cold and hot conditions. Hence, they are used in mayonnaises and salad dressing. Esterified starches have lower gelatinization temperature and retrogradation, lower tendency to form gels and higher paste clarity, and are used in refrigerated and frozen foods as emulsion stabilizers and for encapsulation of beverage clouds. Now continuing the function of starch, pasta, spaghetti, macaroni. Pre-gelatinized and crossing starches are mostly used in pastas. Gelatinized starch affects pasta's elasticity and softness, delectableness and digestibility. Puddings, for example custard. Pre-gelatinized starches are used in puddings, instant lactic mixtures and breakfast foods to achieve thickening or water retention without applying heat. They are also used in ready-to-use bread mixtures. They are used where little or no heat is required, and the increased absorption and retention of water improves the quality of the product. For example, used in meat industry or filling for fruit pies. What is glycogen? Glycogen is a carbohydrate and the storage form of glucose. It is a polysaccharide. Its structure is similar to amylopectin. Glycogen is a highly branched chain of glucose synthesized by animals for the purpose of glucose storage in muscles and the liver. It acts as a secondary long-term energy storage. Muscle glycogen is quickly converted into glucose by muscle cells for energy, and liver glycogen that converts into glucose for use throughout the body, which include the central nervous system. Glycogen does not exist in plant tissue. It is highly concentrated in the liver. Although skeletal muscles contain the most glycogen by weight, it is also present in the lower levels in other tissues such as kidney, heart and brain. Glycogen is a long polymer that branches about every 10 glucose residues. Glucose contains two types of bonds in glycogen. Number one is alpha 1,4 glycosidic bond. Number second is alpha 1,6 glycosidic bond. Cells such as hepatocytes in the liver cells can use glycogen to regulate the blood glucose levels in the body. Now continuing the glycogen, the alpha 1,4 and alpha 1,6 glycosidic bonds in linear strands and at junction points respectively. The branching is a crucial aspect of glycogen as it increases its solubility and allows it to get metabolized more quickly. Importantly, glycogen serves to maintain glucose homeostasis in the animal body. Because of this, its metabolism is regulated primarily by insulin and glucagon and molecules in their downstream signaling pathways. Insulin and glucagon promote glycogen synthesis and breakdown respectively. Brain cells depend almost solely on glucose for energy. Our body also depends on glycogen breakdown during periods of rapid activity such as sprinting. The glucose obtained from glycogen can be used to produce energy under anaerobic conditions in which no oxygen is used. Glycogen is either synthesized or breakdown depending on the needs of the body. It is an essential molecule for maintaining glucose homeostasis in the body. Two major peptide hormones involved in its regulation are insulin and glucagon, which promotes anabolism and catabolism of glycogen metabolism respectively. Here we see the structure of glycogen, in which 1,4 linkage and 1,6 linkage of glucose units are available. Now the breakdown of glycogen. The breakdown process of glycogen consists of 3 steps and uses of 4 different enzymes. Step 1, phosphorylases. Glycogen phosphorylase, the key enzyme in glycogen breakdown, is the first step of glycogen degradation. It cleaves the alpha-1,4-glycosidic bond between a terminal glucose residue with a free OH group on the fourth carbon and the adjacent glucose by using an orthophosphate. It cleaves its substrate by the addition of orthophosphate to yield glucose 1-phosphate. The cleavage of a bond by the addition of orthophosphate is referred to as phosphorylasis. Now continuing the breakdown of glycogen, step 2, which is also known as glycogen remodeling. Glycogen phosphorylase can only cleave alpha-1,4-glycosidic bonds. It does not cleave alpha-1,6-glycosidic bond. In fact, the enzyme stops 4 residues from the nearest branch point. In order for glycogen breakdown to continue, the glycogen must be modified. 2 different enzymes, transferase and alpha-1,6-glycosidase, are responsible for the process. Now continuing the breakdown of glycogen, 2 enzymes are used, transferase and 1 alpha-1,6-glycosidase. Transferase, which is an enzyme, catalyzes the shift of 3 glucose residues from one branch to the others. This process leaves a glucose that is linked via an alpha-1,6-glycosidic bond. Alpha-1,6-glycosidase then cleaves the alpha-1,6-glycosidic bond to release a free glucose and forms the modified glycogen. Notice that the branch glycogen is converted into linear polymer. Now the last step is the step 3, glucose 1-phosphate conversion. The final step is catalyzed by phosphoglucomutase. This enzyme contains a catalytic residue that ultimately transfers the phosphate group from carbon 1 to carbon 6, and the glycogen molecule splits. What are the functions of glycogen? Glucose is stored in the liver and muscle cells. Hydrolysis of glycogen releases glucose when the demand for sugar increases and then burned for calories, providing energy. Glycogen helps maintain hemostasis by releasing sugar into the bloodstream if needed. Some of the glucose can be converted back to glycogen to save for later. Glycogen maintains blood glucose levels. Liver glycogen acts as glucose reserve hepatocyte, which release when there is a need to maintain a normal blood sugar levels. Muscle glycogen serves as a fuel reserved for supply of ATP during muscle contraction. Glucose from glycogen stores remains within the cells in skeletal and cardiac muscles and is used as an energy source from muscle work. Now continuing the functions of glycogen. Brain includes a small amount of glycogen in astrocytes. It gets accumulated during sleep and is mobilized upon walking. Glycogen reserves also assure a moderate degree of protection against hypoglycemia. It has a specialized role in fatal lung type 2 pulmonary cells. These cells start to accumulate glycogen at about 26 weeks of gestation and then synthesize pulmonary surfactant. Thank you very much.